So now our next speaker is Bill Kurz. He is the principal software engineer, Janelia Research Campus. Bill has worked on data management for large-scale brain reconstructions for over a decade at Janelia. He has developed a connect-to-mix data platform for large brain data reconstruction. He also worked on deep learning and sparse volume visualization. In the past, he has been involved at Stanford BioX and was a founding engineer of an image-focused software startup, later acquired by Varian Medical Systems. So today, he will be talking on the management of image-based reconstruction data at Janelia.
Hi, thank you very much. So I'm going to be talking about just a small portion of what Janelia does. It's a very large research center which has lots of different types of imaging. And in particular, I'm going to be talking about the tools that we've created in the connectomics field, which is to reconstruct brains from very large scale EM images. And so this will be, very high level. And if any of this stuff sounds interesting, I suggest that you reach out to me via email or just grab me in the next couple of days, and I can refer you to individuals of the stuff I'll be talking about, or we can talk a little bit more. And so what we typically start off with is we have a small number of really, really large EM image volumes. And so for us, right now we're running about like 50 teravoxels is how we would describe it. And so one grayscale volume could take us years in order to do reconstruction. So there's a tremendous amount of curation, manual curation, as well as automatic artificial intelligence curation before we even start using proofreading. So just in terms of the fly brain, Janelia has put out a whole number of EM volumes, each of which, as we've reconstructed it, we've published it into papers, as well as published the data behind the papers. There's a serial section, very large scale TEM. It's been used as the basis of the most recent full CNS reconstruction. And then there's a whole series of FibSem data sets, of which we are currently also have a male, very large CNS that we're in the process of reconstructing. Now, just getting from these initial images to where we start doing proofreading and automatic segmentation requires a lot of effort. And so I'm just going to briefly describe, some of this initial stages, which would be, going through all the data, figuring out where there are errors, figuring out where there are artifacts, how to look at all this stuff. One interesting aspect here is that the people that are involved with doing this, they found that GitHub is very appropriate, that you can actually do issues, you can track things. So it's very useful in terms of setting up a pipeline and then labeling what kind of problems might be occurring. So that might be useful to you. Or we can also, put you in contact with the people that have set up a methodology for this. There's a tremendous amount of pipelines, very large, more or less Java-oriented pipelines that run on our cluster that handle all of the alignment, the transformations necessary to bring in a variety of different types of large-scale 3D EM images into one sort of consistent volume where we have a single voxel represents certain grayscale value, and eventually will be identified as a label. Like what we would eventually want to get to is in this massive 50 teravoxel volume or much, much higher. Each voxel in this volume has a unique UINT64, 64-bit identifier saying this is part of this neuron, or this is a part of something else that we're interested in. And so we collaborate with Google AI in order to do this initial stages, and so they can spend massive amounts of money doing this and with all of their clusters. And then we'll also supplement this with information on our synapses. And if we zoom in a little bit more, you can kind of see that, that each of the voxels are pretty well differentiated in terms of where the neurons are and what this is sort of pseudo coloring, but each of these different colors represents a unique integer that represents a three-dimensional structure. Now, one of the ways that we kind of hold all of this data is that we first break up the labels into sort of smaller components, things that we can be pretty conservatively say these voxels should be grouped together, and we call these supervoxels. And then what we provide is a way to very quickly link these supervoxels together to create real biological structures. And so these would be like the agglomerated labels here. You can see they're a little bit more fleshed in, look a little more neuron-like. And then in between it is this mapping of the labels from this initial supervoxel conservative assessment to what we really want to get at. And from a data perspective, these supervoxels are mostly static. And so we can represent them in a fairly efficient way. So they're not constantly mutating under the thing. We don't need a database for this. We can use something simpler. And then for the ones that we eventually get them all together, there may be edits, these processes, this proofreading goes on over many years. And so we need to be able to track all the changes that are being done by people, by programs, by a variety of people that are experts in particular regions. And then the one way to look at how to structure the data is that in this massive volume, we break it up into smaller chunks. And this is why, we eventually use for particularly the static components already these, these file formats like XAR N5, NeuralGlance pre-computed. These are all stored in things like AWS S3, Google Cloud storage systems. And we can get at these very quickly if you already know the volume. Now, the problem is that you may not know exactly where, a neuron will stretch. So in this particular case, this neuron goes across the entire brain, which is a massive amount of volume. And so you won't be able to easily say, okay, I want just this neuron and not everything else around it. So how would you wind up doing that? Well, this is an example of something that we want to do in proofreading, which is, we have these more conservative segments, but really they form one neuron. We want to say they connect together. And so we need to very quickly show these to humans and say, should these be together or should these not be together? And here's an example of after all this proofreading, we just released this optic lobe, a paper as well as the data set that we published. And what you're seeing here is really one mesh for every single neuron that is represented by all the voxels. So really, there's many different ways to represent a neuron, one that's very based locally or one that's a mesh where you can kind of show the whole image no matter how it spans. In addition to this, we also have things like we're getting synapses. And so on the left is the grayscale as we're scrolling through, and on the right, you're going to see artificial intelligence that's been applied to figure out where the synapses are. And here we have the region of information, sort of the sectors of the brain that are corresponding to the different parts of the fly brain. So how does all this get held? And how do we actually do this data management? And obviously don't have enough time to go into all of this, but I can give you a very high level picture, which is like the grayscale, the static information, many cases, the supervoxels that don't change that much. They get stored in these cloud object stores like AWS 3, Google Cloud Storage, and this is petascale, so it can handle any amount of volume. In terms of connectomics, we create this layer, which is HTTP API service. So it's a system that operates at a very high level. And we've got two versions of this. One is one we did within Janelia, which operates, maybe up to about 50 to 100 teravoxels. So, in the terms of connectomics, reasonable sized data sets, but not like the extreme high, very, very large ones, and certainly not when we go to mouse or eventually the human. And then the cave system, which we're also starting to use at Janelia, which is, Princeton and Allen Institute and various other places where for this you need the whole Google Cloud platform. It's petascale, use Kubernetes. It's much more involved in terms of the DevOps stuff. The interesting thing that is that for both of these systems, there is this concept of versioning. We do years of curation. And so we want to keep track of what happens, and we can do this by keeping control of the deltas. So we don't have to keep, make copies of every single version as we go along. So we have quite sophisticated ways of doing the versioning, which include ways that kind of mirror almost like GitHub, where you have branch versioning so that you can try different ideas. And it's still kind of an evolving research area. And this is an example of like how something that's like this might happen. You have a science sort of connectomics level API. This is all HTTP API calls like you would with a web. And in the bottom back end, it gets distributed onto a whole variety of storage systems. And let's see if we can, in a little bit of time here, to show you what this kind of looks like. And it looks like it reset. So this is another reason why you never do live Q&As with the thing here, but maybe I'll just burn some of this Q&A time and see. Okay. So what you're seeing here is some of the data is coming from one server in Virginia, which is the segmentation, and some of the other data is coming from a Google Cloud platform, which is the grayscale information. I can, kind of zoom in here. I can zoom out. Some aspects of this are, if I can actually control this a little bit better here, is that, I can click one of these things here and another neuron might come up or two, like this. And then you can kind of see here that, let me change the coloring so you can see the differences. They're kind of coming in layered here. And then this system that you see here is called NeuroGlancer. It's open source. All the stuff that we're showing here is open source. It's a very nice way of scrolling through massive amounts of data. And you can, looks like I lost something there. Yeah, okay. So it's a great way of showing a lot of data. And back on the other side. And then once we get through with our, the reconstruction process, we want to publish all this. We typically take all of that reconstruction service, which has all the versioning, and then we export more static files. An example would be the optic lobe stuff I just showed you, where it's just all the data sitting in a Google bucket in a variety of relatively open format, output for skeletons and meshes and three-dimensional volumes. And with that, I'd just like to acknowledge, the stuff that I've been describing is a lot of people's work across the connectomics community. If you're interested in any of the stuff that I talked about, there's like these links here that you can reach, or you can just reach out to me and we can describe it a little bit later. Thanks.